proteins, right? And phosphorylation usually has what kind of effect? An activating effect on the target protein. But the CDK cannot implement its function without the help of a cyclin. So the cyclin must bind to the CDK. Once this complex forms, then this complex goes in and activates target proteins by phosphorylating them. But what does that do, in essence? Look at what's happening in figure 15.4 on page 487. You see that in the center you have the ring, this orange or yellow ring represents the cell cycle. And then you have these bands that are parallel to it, and these bands have increasing widths and decreasing widths. They have a certain width to them, a characteristic width. That width represents the concentration of the cyclin, CDK, the cyclin complex, the cyclin CDK complex. So as you can see, if you start with G1, the concentration of the CDK cyclin complex in the dark green increases gradually until it reaches a certain height and then decreases. So that increase will initially transition from G1 to S. Then in S you'll have another type of a CDK cyclin complex that will be increasing until a certain point, then it rapidly decreases. So as these CDK cyclin complexes are forming, they're phosphorylating their target proteins, causing the cell to transition then into the next phase, from S to G2. And then again you have the same phenomenon which will cause the cell to transition from G2 to N. In other words, the cyclin CDK complexes are responsible for driving the cell cycle. They're responsible for driving the cell cycle, for regulating the cell cycle. How do they do that? Well, the CDK cyclin complexes are responsible for targeting and phosphorylating their target proteins. And that phosphorylation is going to cause the cell to transition from one stage to the next. And we'd like to understand more in detail how that happens. Anybody have any questions about cyclins? Yes, cyclins are proteins whose concentrations fluctuate during the course of the cell cycle, in synchrony with the cell cycle. They fluctuate in synchrony with the cell cycle. Did you get everything there? Did you need it? You have one more? Sure. But you got these? Did you get these? Yes. Okay, great. Fantastic. Thank you for your patience. Would you like to copy these down? Thank you for your patience. Just to prove to you that we actually do something here. Now, you have a question about cyclins. What are cyclins? Cyclins are proteins whose concentration fluctuates in changes in synchrony with the cell cycle. So at certain times, their concentration will be low, but then will increase and then decrease. Increase and then decrease. And each increase-decrease corresponds to one cell stage. And then it goes to the next one. Increase-decrease, increase-decrease. When it reaches its highest concentrations, that's when the concentration of the cyclin CDK complex will be at its highest, and then it decreases when the next cell stage begins, and it'll increase again, then decrease. But the key point to understand is that the CDKs require the cyclins to be at a high enough concentration in order for this complex to form. And what they will do is phosphorylate their target proteins, causing certain changes in the cell to occur. So please be sure to read chapter 15. It's a really well-written chapter. We'd like to now talk about this pathway that we mentioned several times in these questions, the RBE2F pathway that governs the G1 to the S transition. The G1 to S is the most important transition point. It's called the restriction point for the cell. Because what happens in G1? What is happening in G1? In G1, the cell is preparing for the S phase. What's happening in the S phase? DNA replication. the G1 is a very important stage when the cell is making all of the preparations it needs to synthesize DNA in the set S phase. And it needs to be sure, number one, that the DNA is in good shape, meaning that it is free from any dangerous or harmful mutations. So it's the job of several proteins in the G1 to go through the DNA and make sure that there is no significant damage present. Why? Because if you go into the S phase carrying along this damage, you will simply be perpetuating that, and that's not good, right? That's not good. So you don't want to perpetuate. You want to repair the DNA damage before you go to the S phase. Before you go to, sir, did you need those two? Oh, I'm sorry about that. I forgot. One. Yeah. Oh, I'm sorry about that, sir. I forgot about that you needed the questions too. Once she's done, she will give it to you. Thank you for your patience. So do you have those questions with you?
I was just saying I got the questions, so my DNA solid.
Oh, okay. Oh, okay. That's a good idea too, if you can do that. Sir, do you have those questions? Do you have those questions?
Not all.
Not all.
I can answer the last few questions.
Okay. If you need anything, please let me know. Okay, great. Thank you, sir. Appreciate your help. So remember, the G1 to the S phase is a very important restriction point. So this pathway explains what's happening. Look at this pathway. What's happening here? So there are two proteins that are paired together, the RB and the E2F. The RB is basically the banana-shaped protein, and the E2F is the more egg-shaped protein embedded inside the RB. The E2F is a very important transcription factor. What it's going to be responsible for doing is activating those genes responsible for basically promoting or implementing the replication of the DNA in the S phase. In the beginning, what the job of the RB is to sequester the E2F, to hold it, to inhibit it, to prevent it from doing anything. When the CDK cyclin complex forms during late G1, when its concentration reaches a sufficient point, then what the CDK cyclin complex does is that it phosphorylates RB. It phosphorylates RB. What effect will that have, the phosphorylation on RB? It causes a conformational change in the RB. It causes a conformational change in the RB that causes it to detach from the E2F, releasing E2F, and then E2F is free to do what? Exactly. It's free to activate the genes that are responsible for DNA synthesis. However, we will only be allowing this to happen if there is no significant DNA damage present. So, let's go to a next picture, figure 15.8 on page 491. Figure 15.8 on page 491. This is also very important. It's talking about here is that what if there is some DNA damage present? We don't want that E2F to be free. Because if it's free, then there's nothing to stop it from activating the genes responsible for DNA synthesis. So how are we going to prevent it? Well, what the DNA damage does is that it is detected by a special protein called P53. P53. P53 is known as the guardian of the genome. It's known as the guardian of the genome. And what it is responsible for doing is reading through the DNA and looking for any significant damage. If it encounters the DNA damage, it then triggers the production of another protein called P21. And what P21 does is that it will act as an inhibitor and prevent the CDK cyclin complex from phosphorylating RB. What effect will that have? If the CDK cyclin complex is prevented from phosphorylating RB, what effect will that have? E2F will not be freed, right? E2F will not be freed. If E2F is not freed, then will the cell go from G1 to S? It will not be able to, right? because the E2 has not been freed. So that's the goal of P53 and P21. So please be sure to understand these two figures. Figure 15.5 and 15.8.